Hi, my name is Christian Dallago. I am at Lab at the Technical University of Munich, and today I'm going to talk to you about bioembeddings. Bioembeddings is a modular Python pipeline for protein feature extraction, visualization, and prediction. We extract features using language models, which are generally used for natural language processing approaches, like to translate from one language into another one. Over the last year and a half, and even more, in protein bioinformatics, language models have been used for various purposes. For generative purposes, to, for example, create new protein sequences from a set of protein sequences which you are interested in and have a particular feature which you want to emulate. To predictive purposes, for example, you want to predict the subcellular localization from just a protein sequence, or you want to annotate each amino acid in a protein sequence with a secondary structure. So since there are so many language models already out there over the last year and a half, and probably many more to come, we wanted to just abstract the use of these language models for researchers out there. So that's why we came up with bioembeddings, which is just a pip installable package. You pip install bioembeddings. You then collect your sequences into a FASTA file. And you lastly have to define a configuration file which allows you to reproduce your pipeline run in the future should you want to publish your results and want the results to be reproducible by other labs. We also allow you to use a default configuration file which basically runs 3 stages. What the stages are we will see in a second. It will run embed, project, and visualize. And from the output of this default pipeline run, you will be able to explore the embeddings using Jupyter Notebooks, which we also make available online. You will be able to use the embeddings as features for your prediction tasks, and there's an example of that online as well. And from the visualize stage, you will get a scatter plot of your input sequences, which you can use to see if the language model that you have chosen is able to separate well the sequences that you have inputted. So onto the stages which are currently implemented in the pipeline. And there are 3, with the 4th one coming soon. Probably the most important stage is the embed stage. This takes your input sequence FASTA and embeds every sequence in the FASTA file with a particular language model of your choice. The language models currently available are 2. There's a LSTM-based model called SecVec and a transformer-based model called Albert. We are in talks with other labs to include models coming from other labs, as well as developing our own models for just to expand the diversity of models which we can offer through bioembeddings. The project stage takes these embeddings, which are usually very highly dimensional, and projects them onto lower dimensional spaces using popular techniques like UMAP and TSNE. And these lower dimensional representations of your input sequences can then be used for analytical purposes and for visualizations. So the visualize stage takes these lower dimensional representations of your sequences and displays them in a scatter plot, which can additionally be annotated with an annotation file which has labels for some or all of the sequences in your set. Another thing which we want to offer with the visualize stage is the visualization of the importance of each amino acid in the input sequence. And we will offer this for models that use attention layers. The last stage, which we already are using internally and we will make available publicly soon, is the extract stage. And this stage comes into 2 flavors. There's the supervised version where a particular machine learning device has been trained on a labeled data set and labeled embeddings to predict, for example, secondary structure for each amino acid in your sequence, or subcellular localization for each sequence in your set, as well as an unsupervised version of extract, which looks at the closest hit in embedding space to a certain labeled data set. So let's suppose you have embedded all of SwissProt with annotations of Go, and you have a set of sequences which are unannotated. So you just look at the closest hit in SwissProt to your embedding, and then you just transfer the annotations from that closest hit onto the sequence which you're currently looking at. So this is the supervised and unsupervised extract stages, which are coming soon to the pipeline. Just to give you 2 examples of the use case, 2 use cases. One is I want to visualize how well a language model separates my data, which in this case is labeled. So I took the example of CAF and Disprot, both databases for structural features, one secondary structure and the other one disorder. So what we can see is that by using a language model, in this particular case SecVec, the language model is already able to, without having been trained on distinguishing mainly alpha, mainly beta, or little disorder, mostly disordered, it's already able to distinguish these two groups to some extent. So probably using this language model and the extracted features from this language model is a good input to train a machine learning device for the classification of these data sets. Another example is the example of the transfer of annotations from a labeled data source to sequences which you are currently analyzing. And in this particular case, it's using embeddings to transfer Go annotations from Swissprot onto to an unlabeled dataset. And for this particular example, I invite you to listen to the talks by my colleagues who are also presenting at ISMB this year, Maria Littmann and Michael Heinziger, for which you find the title here. So to conclude, we have tried to develop a pipeline which allows you to go from faster sequences to features using language models, and then to just use these features for predictive, analytical, generative purposes as you see fit. We try to include as many language models as possible, and we're constantly improving both the pipeline as well as the extent to which it works. If you have any questions, just shoot me an email or a tweet, and you can also open up issues on the GitHub repository, which is linked below. Thank you very much.